Okay, so now let's learn about try solving some problems in genetics. We're going to work on some solving problems dealing with chromosomal variations or abnormalities. We're going to look at situations in which an individual may gain or lose an extra chromosome, or gain a chromosome or lose a chromosome, as well as have different types of rearrangements of the chromosomes that they have. So for this first problem, I want to look at look at a situation in which a child has an extra chromosome, and in this case, an X chromosome. Now, this child has mild intellectual disability and otherwise is phenotypically a normal boy until he reaches puberty, at which point he fails to develop normal sexual male development and begins to develop some breasts, which is characteristic of a female, and so his parents bring him for some genetic testing. And what they find is that the child has an extra X chromosome when they do karyotype analysis. So the first question is, what is this boy's genotype for the sex chromosome and his karyotype? And his karyotype. And so his genotype would be 47 XXY, because a normal male would be 46 XY, and this individual has an extra X chromosome, so it'll be 47 XXY. And so and then the next question is, what is the genetic condition described by this boy's sex chromosome composition? Well, this condition is what we refer to as Klinefelter syndrome. Klinefelter syndrome. And what genetic event is responsible for this boy's unusual genotype? And well, this is when we have an extra chromosome or we're missing a chromosome, we refer to that individual as being an aneuploid or aneuploidy. Is the condition that is an aneuploidy, and the genetic event that's responsible for this is from when we fail to separate either the sister chromatids during meiosis or homologous pairs during meiosis. That results in having a gamete that has an extra chromosome, and so this failure of properly segregating our chromosomes is referred to as non-disjunction. So now the next set of questions that we're going to look at. So now the genetic counselor tells the parents that this is the most common type of an aneuploidy. Why do you think this is the case? And what mechanisms help accommodate this abnormal karyotype? And so we do see, in terms of aneuploidies, where we have the incorrect number of total chromosomes in an individual, that most frequently this is going to be found dealing with the sex chromosomes, either having an extra X chromosome, as the case of Klinefelter syndrome, or an individual that may have Turner syndrome, in which they have just a single X chromosome and no other sex chromosome. The reason that these are the most common aneuploidies that we will find is that because of the fact that mammals, humans being mammals, have an imbalanced sex chromosome system between males and females, in the sense that males have an X and a Y while females have two X's. And so this imbalance, or this this difference in total chromosome number between males and females, means that we need to have some mechanism in place that's going to allow for changing expression of the genes on one of these chromosomes. So we could do one of two things. Is, since females have two X chromosomes, we could try to increase the level of expression of the genes on the X chromosome in the male, or alternatively, we could reduce the amount of expression in the female. And so mammals do that latter thing, in which they will reduce the amount of expression of the X chromosomes. And the way that they accomplish this is through X inactivation. And so X inactivation is something that happens in all cells within females, in which one of the X chromosomes is randomly inactivated, so it's therefore referred to as random X inactivation. And so this random X inactivation happens, in which that will reduce the amount of expression for most of the genes. There is a little bit of expression that does happen off of the inactivated X, which is why an individual with Turner syndrome isn't phenotypically normal. They're more apt to have congenital heart defect. They may have some other physical developmental differences that what can account for that partially would be the fact that that inactivated X in a karyotypically normal female isn't completely inactive. It is essentially inactive. But because we have this dosage compensation mechanism already evolved and in place within females, we're able to tolerate and deal with differences in chromosome content with our sex chromosomes better than what we can with our autosomes, or those chromosomes that we number 1 through 22 that everybody has 2 copies of. All right, so now the mechanism that's in place to accommodate for this would be random X inactivation, and it's because of the fact that we have this dosage compensation mechanism in place. All right. Now the next question we need to can sometimes be a little confusing, and people often struggle with trying to understand how this question works. And so here we have a situation where we're given the genotypes of parents. We're given the genotypes of an aneuploid offspring. And what the question is asking is where did the non-disjunction event occur? So when did non-disjunction happen? Did it happen during meiosis 1 or meiosis 2? Or can we not determine? And did it happen within the mother or within the father? Okay. So in order to better understand this sort of a problem, I'm going to draw out a little bit of the difference between non-disjunction in meiosis 1 versus non-disjunction in meiosis 2. And so if we move this out of the way. And so when we have meiosis 1, so remember in meiosis 1, during meiosis 1, this is the reduction stage of meiosis, in which we're reducing the amount of chromosomes to half. So all cells within a human have 46 chromosomes typically, in a karyotypically normal human. And when we go through mitosis, which where we're just creating a new cell that's identical to the parent cell, we're going to copy our DNA, all 46 of those chromosomes, and divide one copy of each of those chromosomes into each resulting daughter cell. But when we're doing meiosis and we're producing gametes, we need to reduce the amount of chromosomes we have from 46 to half, which is 23. So we need to go from the diploid state of 46 to the haploid state of 23. Now, the way that this is going to be accomplished is we're going to go, rather than in mitosis, we duplicate the DNA. We do one cell division and divide that DNA equally into each daughter cell. In meiosis, we're going to duplicate the DNA, and then we're going to do 2 cell divisions. And in the first cell division is where we're going to reduce the chromosome content from 46 chromosomes to 43 chromosomes. And we're going to accomplish that by separating homologous pairs. Then when we go through meiosis 2, what we'll then be able to do, or what we will then do is separate our sister chromatids of all of the chromosomes that are left in each of those resulting gametes. And so if we have a cell, for instance, and say we have, we have a cell, and here, and for this case, I'm just going to draw X chromosomes, okay, and I'm going to indicate with an M or a P, we're going to say XM is the maternal chromosome, and XP is the paternal chromosome. Now, maternal is referring to the X chromosome that this individual received from their mother. Paternal chromosome is the X chromosome that this individual received from their father. So when we go into meiosis 1 and we're looking at metaphase 1, phase 1, we're going to have our homologous pairs align along the metaphase plate where they're paired like this. So we'd have an XM on one side and an XP. And so during normal cell division, we're going to separate our homologous pairs. So we're going to divide this cell in half. Okay, and when we divide this cell in half, we're going to pull the maternal chromosome apart from the paternal chromosome. And so during normal cell division, and I'm just going to draw this going into straight into metaphase 2. Okay, we'd go through anaphase, we'd go through interphase, and then we would recondense and go into metaphase 2. And then during metaphase 2, we would have the XM chromosome on this side, the XP chromosome down this side. Then each of these in meiosis 2, we're then going to separate the chromosomes by the sister chromatids and result in 2 gametes with a chromosome from the maternal side and 2 gametes with paternal X chromosomes. So we would separate our sister chromatids, and we'd end up with a cell here that's XM, and we'd end up with a cell here that's XM. And then here we would end up with a cell XP and another cell XP. Okay, so that's what happens during normal cell division. Now, when we look at non-disjunction, and that non-disjunction event occurs either in meiosis or meiosis 1 or meiosis 2, if it occurs in meiosis 1, it means that we're going to fail to separate these homologous pairs, and they're both going to go to the same side and go to one daughter cell, and the other daughter cell is going to have none. If we have non-disjunction happen within meiosis 2, then we're going to fail to, in one of these 2 daughter cells here that we have, we're going to fail to separate our sister chromatids, and they're both going to go to one of the daughter cells that would be down here, and one's going to lose it. So will then be lacking a chromosome. And so if we have non-disjunction in meiosis 1, what we will have is our chromosomes aligned along the metaphase plate just as they were before. Okay, where we have XM and XP. And instead of them both going to each respective side and to each new daughter cell, we could have both of them go to one side, in which we fail to segregate or separate these homologous pairs into their own daughter cell. In which case, when we get to, and this would be in meta 1, and this would be in meta 2, and so when we're looking at our daughter cells in metaphase 2, we're going to have 2 X chromosomes, an XM and an XP in that daughter cell, and we're going to have no X chromosomes in the other daughter cell. So now when we proceed through meiosis 2, presumably we're going to only have a single meiotic non-disjunction occur within any round of meiosis. If we have more than one loss or gain of chromosomes, likely that would result in a non-viable offspring. So we typically aren't going to see that. And so when we go through in this cell that has this non-disjunction during meiosis 1, most likely we're going to have normal cell division proceed through meiosis 2, and if we don't, we won't actually observe an individual that from that resulting gamete, so we wouldn't actually see it. And so here, this cell is going to divide into its respective daughter cells, and now instead of a single X chromosome, like we see here, 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 and here, we're going to have 2 X chromosomes in each of these daughter cells, rather than 1. An XM and an XP. An XM and an XP. Okay. And then these, in terms of sex chromosomes, has no sex chromosomes. So when we divide that all of the other autosomes will divide normally, and the sex chromosomes, there aren't any to divide. So in terms of sex chromosomes, these daughter cells will be missing one. And so these will be lacking a gamete, or lacking a sex chromosome, and these will be lacking a sex chromosome, and these will have an extra sex chromosome. Okay, so now the key thing to look at here is, so when we have non-disjunction that occurs within meiosis 1, we now have 4 gametes that result from the cell division that are abnormal in their chromosome content. We have 2 of the gametes have an extra chromosome. 2 of the gametes are missing a chromosome. Okay. The other thing to take note of is the fact that the 2 gametes that have extra chromosomes, they have 2 different types of chromosomes. Here we have an XM, a maternal, and an XP, a paternal X chromosome, and an XM and an XP. Okay, and so that's to contrast with when we look at non-disjunction and how it occurs. Oops, sorry about that. Non-disjunction and how it occurs in a cell during meiosis 2. And so we have non-disjunction in meiosis 2. If we look at metaphase 1 again, okay, we look at metaphase 1. Here we have the same arrangement that we looked at before, which we have an XM and an XP. So this is non-disjunction occurring in meiosis 2. So meiosis 1 proceeds normally. So we separate our homologous pairs into the daughter cells. And so if we look at metaphase 2 of meiosis 2, we're going to have an XM here and an XP here. And so when we go through this next round of cell division of meiosis 2, we're going to have a single meiotic non-disjunction. We'll assume for the sake of the example. And so one of these we're going to fail to separate our sister chromatids, and the other one will presumably proceed normally. And so if this one proceeds normally, what we'll end up with 2 gametes here that contain a single X chromosome, so are normal. And so in this case, rather than both of those sister chromatids going to the 2 new daughter cells, they both go to the same side. And so we would end up with a single daughter cell here that's lacking a chromosome, and this daughter cell that has 2 X chromosomes. But different than non-disjunction in meiosis 1, when we had non-disjunction in meiosis 1, we have an XM and an XP. We have these gametes have an extra X chromosome, but they're a maternal and a paternal X chromosome. When we have non-disjunction in meiosis 2, here we have 2 X chromosomes, but they're both paternal and paternal. Okay, so they're the same exact identical chromosomes. So that's an important distinction. And the other thing to note when we're looking at non-disjunction in meiosis 2 is here we have 2 normal gametes, and then we have 1 gamete that is lacking a chromosome, that's missing a chromosome, and 1 gamete with an extra chromosome. And so within this meiotic event, we have 4 gametes that result. 2 are going to be normal, 2 are going to result in an aneuploidy. And so if a normal gamete fertilizes, say if these are eggs, these would be eggs considering they're They started out as an XX individual. So if this was an egg and gets fertilized by a normal sperm, okay, if this is a normal sperm, that individual would be a trisomy individual, or have an extra chromosome. And if the normal sperm fertilizes this egg, it would be a monosomy, such as Turner syndrome, if the X fertilized that. Whereas these individuals, the normal sperm would then just produce a normal individual. Whereas with the non-disjunction during meiosis 1, all 4 gametes are abnormal in their chromosome content. Right, so now, keeping this in mind, this is what's going to make it useful to be able to answer these questions. Okay, so when we're looking at this question, so we're looking at a situation where the gene for red-green color blindness is found on the X chromosome. Now, this gene has 2 different alleles, where the recessive allele is what gives you color blindness, and the dominant allele is normal color vision. And so here we have the presence of a single copy. This big RG is enough to give you color vision, and the little RG does not encode for color vision. So now when we're looking at these individuals here, we have a father, an XY individual, that is having a child with a woman who's colorblind because she has little RG, which is going to tell us that she's homozygous for the recessive state of colorblindness. And they have a child that is XXY. Now, given the allelic makeup of this child, we can, one, say that this child has Klinefelter syndrome because this child's an XXY individual. Now, the question, though, is asking, did the non-disjunction occur in the mother or the father? And did it happen during meiosis 1, meiosis 2, or can we just not tell? Okay, and so in this case, what you want to, for a problem like this, what you want to do is you look at the offspring and you have to try to figure out where each of the chromosomes have come from. And so the alleles that are on the X chromosomes is what's going to guide our thinking and where those chromosomes came from. So here we have and remember, each parent should be only contributing a single X chromosome, or a single chromosome, or sex chromosome at all. And so here this individual has an X big RG, and so they got that from the father. Here's an X little RG, they must have gotten from the mother. And then the Y, so they got that from the father. Okay, so now this tells us one thing that we can say is, well, this must be from the father. And the reason we can say that is because the individual has 2 chromosomes that came from the father, which means that non-disjunction must have happened in the father. Okay. Now the next question is did it happen in meiosis 1 or in meiosis 2? And for that, we need to consider our situation over here, where if instead of calling this the XP, and instead we call it Y, versus we can see our answer. Okay, so this individual, non-disjunction happened in the father, and then this child has an X and a Y from the father, and so if we had non-disjunction in the father, okay, if it happened in meiosis 2, it would either be 2 Y chromosomes or it'd be 2 X chromosomes that are paternal in origin. Okay, but we only have 1 X chromosome that's paternal in origin, and so it must be meiosis 1, in which case that the father failed to separate homologous pairs, or the X and the Y, and then in meiosis 2 resulted in 2 gametes, one with an X, one with a Y. Okay, so that's how we would go about answering that question. Now, so this must be meiosis 1. And the key to this is recognizing that when you have non-disjunction in meiosis 1, that the 2 chromosomes that you have from that single parent are different chromosomes. Okay, contrasting with meiosis 2, they would be identical. Okay, so if this was an individual that was X little rg, X little rg, well, then we would say, well, it was meiosis 2 in the mother, because we would know that the little rg's only could have come from the mother, and the fact that they're identical to each other tells us that it happened in meiosis 2. Okay. So now this next problem, what we're looking at is, again, here we have the same setup of the parents, but now these parents are this offspring. Here we have a X big RG and no other chromosomes. So this is a monosomy. This would be a Turner's individual that has Turner syndrome because they're XO, meaning that they're lacking a second sex chromosome. But now we have a big RG, which we see here that the father can contribute. So we know that the non-disjunction had to have happened in the mother. Okay, so for this, it had to have happened in the mother. Now the question is did it happen in meiosis 1 or did it happen in meiosis 2? Well, in this situation, the mother is contributing no chromosomes. We know it had to have happened in her because we have 1 chromosome from the dad. Okay, but the mother is contributing no chromosomes. And so when we look at our drawings over here again, what we can see is that when we have non-disjunction occur in meiosis 1, we have a situation where we have abnormal gametes that are lacking a chromosome. Okay, so it could have been one of these 2 gametes that got fertilized. But when we have non-disjunction in meiosis 2, we also have a gamete that is lacking a chromosome. And so when we're lacking a chromosome like this, we can't tell which event, when did non-disjunction occur, because in both situations we have gametes that could result in missing a chromosome, and so this cannot be determined. Okay. Now, the question is, how many bar bodies would you expect? So bar bodies, this is a simple genetic test that we can do on an individual, in which when a karyotypically normal female inactivates one of her X chromosomes, that inactivated X chromosome condenses to the side of the cell and is seen on interphase cells as this kind of lump on the side of the nucleus, and that's what we refer to as a bar body. And that happens to any chromosome, any X chromosomes that greater than 1. So if you have 1 X chromosome, you have no bar bodies. If you have 2 X chromosomes, you have 1 bar body. If you have 3 X chromosomes, you're going to have 2 bar bodies. Okay, any X chromosomes greater than 1, you end up having a bar body on the side of the cell. And so Klinefelter's individuals, because they have the Y chromosome, they're typically phenotypically male. But if you do a bar body test, they have 2 X chromosomes. So these we would expect to have a single bar body because they have an extra X chromosome. Now Turner individuals, because they're XO, meaning they just have a sex chromosome X and no other sex chromosomes, they're phenotypically female. But because they only have a single X chromosome, means that they do not have any bar bodies. So we'd expect none. Okay, so that's how we solve those types of problems about non-disjunction, and that's where we'll stop this video. So if you found this helpful, by all means, I hope you follow along and see some of the other videos that I come out with. I'm going to try to work through a bunch of these different types of problems. If you found it helpful, I'd appreciate if you gave it a like so that I know that it's actually helping somebody. And I'm going to continue trying to put some content out there that will help people figure out genetic problems and learn genetics and biology in general. So thanks for your time.